student sections A and B. Today's session will focus on a new topic, which is actually the gene or unit of genetic information. So basically, I'm going to start discussing a very nice experimentation. Hopefully, you're going to like it. Under the title of transgenesis or genetic engineering. So actually, if we have a look at this experimentation, you can see here in red a gene which has been isolated, and this gene is simply cleaved or cut by special scissors and inserted into bacterium, and that gene, which is a fragment of a chromosome, is responsible for the production of what do we call pest-killing protein. So this is a protein that is responsible for killing pests. So it's actually a pesticide. It has been transferred, as you see here in step number 2, to a typical normal cell, a normal plant cell. And by this moment, that plant cell, which has initially its own chromosomes, okay, has right now acquired an additional fragment that doesn't belong to this plant cell because it is originating from a different species. And eventually speaking, we have right now what do we call a cell that is genetically modified. And as the cell is likely to grow under the normal conditions to produce a new plant, the growth of such cell results in the production of a typical corn plant, which is said to be genetically modified. And this genetically modified organism right now is not only resistant to pesticide, but at the same time, it is a factory that produces what do we call pest-killing protein. So this is a very huge advantage. This is a huge business. By the moment we mention that these corn are genetically modified, this is to say that this plant right now is a factory that aims at producing special type of protein, which is a good business. So this is what do we call by transgenesis, or what do we call genetic engineering. So if I want to analyze this experiment in brief, the analysis of this previous experimentation reveals that the grown plant, which is the corn, has become right now a genetically modified organism, GMO, that has acquired a new trait other than having its own plant traits. And this is the trait that I'm talking about, which is the trait of pest-killing protein. Thus, this modified corn plant, known as transgenic plant, has become on one hand resistant to pesticides, and on the other hand an organism that produces such proteins that can be isolated and sold in the market as a product, having both economical and environmental revenues or benefits. So what can we deduce from the analysis of this experimentation? I mean by two notions, two definitions that I'm going to highlight very well in the coming slide. First, the definition of this technique known as genetic engineering, and number two, the definition of a gene, or what do I mean by the notion of a gene. So if you look from left to right, I'm going to start by discussing right now the issue of transgenesis. So if you look at transgenesis, we can see here that transgenesis is the definition that aims at producing what do we call a new organism that is said to be a GMO, and that GMO is simply the result of transferring a specific fragment of a chromosome, which is a gene, and that is cleaved or cut, and transferring this structure from one species to another, as we have seen a few minutes ago. And these are a few examples like the glowing tobacco plants. Can you imagine a tobacco plant that glows at night? Because we have been able simply to transfer a glowing gene from the jellyfish into a tobacco plant. Or some type of tomatoes which are actually ripened, but unfortunately because their texture is very soft and prevents customers from eating them at the proper time, we have been able to produce tomatoes that are very resistant to cold and infection, and in this sense, they become very significant for economy. However, to understand better the definition of a gene, the gene is simply a chromosomal fragment that determines a particular hereditary trait, like the eye color, like the hair color, or the earlobe, whether being free or attached, and the presence of 5 fingers, because some individuals may have 6 fingers. So this video that we have discussed so far aims at introducing the concept of a gene, how we've been able to achieve this concept through an experimentation called transgenesis, and we can have hundreds and thousands of experimentations on YouTube and Google, and the aim is simply unique, where we can see that to prove the relationship between a gene and a trait, we can carry out similar experimentation, and anytime a new organism acquires an additional trait, this proves that this trait is simply represented by a gene that comes initially from such initial cell. Thank you so much and hope to see you soon.